Good morning, class 10th. Children, today we will be completing the remaining experiments of photosynthesis. If you remember, we started with them last time and we got over with, I guess, 2 experiments, but I told you that time too that few are remaining, but we will be getting over with them today only. Right, children? So let us move on with the experiments. The first one here that you can see is to prove that carbon dioxide is essential for photosynthesis. Children, before I move further, let me tell you that all these experiments, I mean your 10th boards paper is all about figure based questions, and you never know plant physiology and animal physiology. I mean the figures from both the systems that you are going to study and the chapters based on plant physiology, they all are equally important. You cannot be one sided at all. So must understand that whatever figures we are showing you, they are actually important. That's why I keep on giving them to be solved in your notebook. So just give them some importance, right, children? So let us start with this. I'm just shifting the camera up. I moved it down to show you that this experiment is based on the essentiality of carbon dioxide for photosynthesis, right? So let us move further. If you can see that I have drawn a stem of the plant, where what are the setups here? Let us understand that if you can see that this is the flask, and inside the flask you can see that I have inserted a complete, I have shown a complete leaf inserted within the flask, and this is the cork. That means the flask is airtight now. You must be wondering again how the leaf got in. So again I am telling you that the cork, which is, yeah, the orange color cork that you are seeing here, we take it out, we slice it into two, half of the slice we put in, then we allow the leaf to enter in, and then remaining half is inserted again, so that the things become airtight and the whole leaf is also inside the flask, right? Another leaf, suppose if we label it as, just hold on, label it as leaf A, and this one is leaf B, right? Because question may be based on the leaves that way, that what is the outcome. And this is the setup, and the blue colored content you can see here, this is the potassium hydroxide. Let me tell you one thing here very clearly. Potassium hydroxide is a carbon dioxide absorber, right? Apart from it, you talk about sodium hydroxide or ammonium hydroxide, these all are carbon dioxide absorbers. But sometimes the question is that if suppose instead of KOH you have put lime water, so will the result be same? In that case, you may go on with the formula and then you see it is also a hydroxide. But children must understand, lime water is something which is used. It is not. It doesn't absorb carbon dioxide on its own. Rather, when you I mean blow the air which crosses this liquid, this say calcium hydroxide or lime water solution, when you blow the air across it, then it turns milky. That means it is an indicator of carbon dioxide but not the absorber. Mind it. And whenever such questions are there that can you use anything, will the setup be giving you a fruitful result? Suppose if lime water is there, then you would say no. And what could be the alternative here? In that case, you will write KOH or NaOH. Right, children? This is just an extra thing because sometimes I have seen that in the figure based questions, such questions are there, they are present. So, take care of that, right? So, again, we are getting back to this setup. If you would have sat in front of me, I would have asked you people that what kind of a plant should I have had taken. So in that case, this is a D-starch plant. I need not to tell you again why we are taking D-starch plant, so that whatever experiment we are doing now, the freshly formed starch is to be tested, not the previously existing starch, right? So we have to remove all the previously existing starch first of all, and then the new fresh photosynthetic product has to be tested, right? So the plant is de-starched as of now. This is one setup, children, and if you can go with the other one, I have drawn it here. Right, this could be another setup where I have not inserted the complete leaf inside. Rather, I have taken, please do not take it as plug. This is just to show you that what could be another setup. This leaf is still intact with the stem. Right, half of the leaf remains out of the flask and half of the leaf I have put in and made the flask airtight, and this is again KOH as a carbon dioxide absorber. Now, if it is a carbon dioxide absorber, that means within the flask there are no traces of carbon dioxide. Keep the plant in sun with such a setup, allow the sunlight to fall on, then allow the plant to perform photosynthesis. After few hours, just pluck off the leaves. This one, for comparison, you can take the leaf A and leaf B, pluck both of them, and then you have to perform the starch test. Now, what is the first step of performing the starch test? You will be boiling the leaf in water. Even you just boil the leaf in water, all the cells are killed, then you boil the leaf in methylated spirit. That means all the chlorophyll is also removed. Now both the leaves are white, pale white. You don't know which one is A and which one is B, right? And then you pour, again you put it in boiled water to make them soft, and then pour few drops of iodine. What will happen? Obviously the leaf which is out, right, it will completely turn blue black, because all the necessary requisitions for photosynthesis were present. But when you talk about B, this leaf will not turn blue black at all, rather it will be having the color of iodine, which is brown, right? It will not turn blue black. Reason behind, because one of the factors which were used as a raw material for photosynthesis, that was carbon dioxide, and carbon dioxide was not present within the flask. So this leaf will not turn blue black, rather it would go with the color of the iodine. Right, children? Similarly, when you go with the second setup, understood that when you take out this leaf and you boil it in water, you boil it in methylated spirit, remove the chlorophyll, it is pale white now, and then you again put it in boiled water to make it soft, and then you pour iodine. What will happen? Half of the leaf which is out, this much only will turn blue black, but the portion of leaf which is within the flask will not turn blue black when you test for starch immediately, right? You are not allowing the starch to move here and there. The mesophyll cells in the leaf which were present inside the flask, they will not produce any starch or any glucose. Why? Because photosynthesis was not occurring there. So half of the leaf blue black, half leaf brown. Right, children? So what is the outcome of this experiment? That if carbon dioxide is missing out of the raw materials, photosynthesis is not going to take place. Okay, so I will be moving on with the next experiment in the next video. If you have any queries regarding this one, just be prepared. I will be there in the problem solving class. You can ask me questions there. You don't utilize your time in those 20 minutes. Actually, they are wasted. Anyways, thank you.